And good afternoon, friends. Morning. Actually, it's 1 o'clock on Monday. And 26th of October. So we had our second exam. I'll get grading on that and get that back to you. We're going to continue on with some discussions about, today, food and waterborne illnesses, things that get into the GI tract. So last time we were covering things that were getting into the respiratory tract. A thing to do, perhaps, while the next exam will still be the same format, but probably a nice idea to help, would be to begin developing a list of the bacterial species and the diseases they cause, and just start to develop a chart, for respiratory tract bacterial diseases, and now GI tract, and then we're going to get to genitourinary tract, and just kind of keep a list going. So today, we're going to be talking about food and waterborne illnesses. And we actually can break this down to a couple of different ways. The GI tract is going to be, bacteria are going to be largely introduced by the things we consume. As we've been saying, we're pretty tough organisms, and about the only way bacteria can get into us is through the various holes we have, or holes you might develop by puncturing yourself with a needle or cutting yourself. But things can get into the GI tract. So you can inhale stuff, but what if you swallow stuff? What are some of the things that can happen to you? And we can break things down in a couple of different ways. This first set of things is what we refer to as intoxications. And these are toxins that you could ingest. And a classic one is a bacterium known as Clostridium botulinum. This is the bacterium that causes botulism, which is not particularly common, but it is incredibly toxic. In the food class that I teach, we talk about foodborne illnesses, and botulism would be kind of a cool thing to talk about. Turns out that it's probably less than 10 cases a year. Botulism is phenomenally rare. One of the things about Clostridium botulinum that's interesting is to note that it is an obligate anaerobe. That means that it grows only in the absence of oxygen, which is, in a food situation, kind of an interesting point. But what can happen is that you can have a canned good, which is sealed up, and a number of interesting things start to happen here. If the canned product has not been sterilized properly, you can end up with bacteria there. Now, the bacteria are going to begin operating and respiring and sucking the oxygen up. The bacteria that are aerobic are going to be the first to start growing, and they will actually scavenge all the oxygen from the can and generate an anaerobic environment, an environment that has no oxygen, and that allows the gram-positive Clostridium to actually begin growing. So it forms endospores, which are really tough, and they're very resistant to a lot of different kinds of heating and things. And so you are cautioned, if you ever do any home canning, to not do your own canning of things that have a neutral pH and things that are really dense, particularly this time of year. Things like pumpkin and squash. Canned pumpkin, canned squash would be an ideal spot for botulism because they're pretty dense material. I mean, like a can of pumpkin, man, that stuff is like packed. And so it's difficult to be sure that you're getting enough sufficient heat all the way through the product to render it sterile. And if you don't render it sterile, you have aerobic bacteria that begin growing pretty quickly. They scavenge all the oxygen up, and then they allow the botulism bacteria to begin thriving. And so warnings would be sort of a bulging can or gases in the can. That's an indication that your can material was either not sterilized properly or something got into it. Botulism, or the Clostridium botulinum, is the source for the toxin known as Botox. We have a picture here of someone getting a Botox injection into the wrinkled forehead. I would say, really ridiculous? Yes. You're putting in a phenomenally potent neurotoxin into your skin. What this does is it blocks the nervous system signals to muscles that cause you to actually have muscle contractions in your face, and it essentially is paralyzing those muscles. It's not permanent. It's probably 6 to 8 weeks or so, and they can actually recover. But I guess vanity, whatever, Let's get over it. Well, that is a real serious one, but not a real common one. So there are a variety of different types of botulism. Wound-associated botulism occurs when you have a deep wound that closes up. So like a puncture wound that closes up, again, you have the bacteria there that are able to scavenge the oxygen in the environment and then establish an anaerobic environment. Infantile botulism, the baby, because the neurotoxic effects on muscles prevent muscle contractions, babies are quite flaccid. Botulism can grow in the gut, and it can be the spores can actually come in through things like honey. You're advised to not give little babies honey because the high concentrations of sugar in honey are osmotically keeping the bacteria from growing, but they can be present as these spores. And raw honey, particularly, is not pasteurized. And then when you ingest it, the osmotic stress is relieved, that is, the sugar is diluted out, and you begin to have the bacteria have the opportunity to start growing. So low acid foods that are improperly canned, that is a problem. Cracked jars, loose or dented lids, probably not the best thing to do. Cooking and reheating foods thoroughly, keep cooked foods hot, or cold foods cold. We're going to see this as a mantra. This is how we can reduce the bacterial load and make it so food is a lot safer. Some of the symptoms, these are things that start to have effects on muscle contractions and muscle tone. So double vision, droopy eyelids, trouble speaking or swallowing, breathing, your muscles begin to lose the ability to contract, and it is going to be fatal if not treated. So it is known as the world's deadliest toxin, and what it's doing is blocking the release of a neurotransmitter. So maybe in A and P you've had a little bit of neurobiology, or if you'll get to this, what we know, or hopefully know, is that nervous system signals are transmitted down a neuron as very, very weak electrical impulses that occur because patches in the membrane undergo what's known as depolarization, a change in the ionic concentrations within a little portion of the membrane that then cause the next set of what are called voltage-gated ion channels to snap open to allow sodium to rush out, and sodium rushes in and changes the charge of the cytoplasm right there and makes it go positive. Well, when you get to the end of a neuron where you have the neuromuscular connection, the neuromuscular junction, the voltage isn't sufficient to actually spark across the gap. What happens is that a series of neurotransmitters are released in vesicles. They fuse with the plasma membrane at the end of the axon, right there. Here, let me get my little red pointer. And they fuse and dump neurotransmitters into this gap called the synapse. And from here, the neurotransmitter binds to a receptor that causes a ion channel to snap open on that side and causes the depolarization, and then that'll actually generate the signal for contraction as calcium is being released through a couple of different mechanisms. Regardless, this is what it looks like. We have the synaptic vesicle in the neuron at the axon tip of the neuron, filled with various neurotransmitters. It fuses with a series of proteins that are on the cell surface, and when it fuses, the membrane is going to actually fuse with the plasma membrane, the membrane of the vesicle, and dump its contents. And the contents then are released into the synaptic cleft. The specific one we're looking at is acetylcholine, which is very common neurotransmitter that is triggering muscle contractions. Acetylcholine binds to its receptor. The receptor opens up, and we have a ion change occur that will trigger muscle contraction. Botulism has a protein that is able to block the targeting protein on the cell surface, the inside of the cell membrane, that the plasma, the vesicle, would bind to. And when it does, the botulism toxin actually inhibits any further fusion of the vesicles, and it prevents the neurotransmitters from actually being able to get dumped into the synaptic cleft. And so even though you have neuronal signals traveling to the muscle, because they can't make that final jump of the neurotransmitters into the synaptic cleft, you don't get any contraction, no signal being done. So it is a very good, very powerful neurotransmitter inhibitor, and it blocks the signals getting to muscle cells. So it is not a very good thing to be messing around with. People do inject it, and what you're doing when you're injecting botulism toxin is you're blocking the signals to those specific muscle cells, and it causes essentially muscle paralysis. So another very common and happily not that deadly problem that we can run into is a bacterium that is on our skin all the time, and that is Staph aureus. Now, it takes a fairly high concentration of Staph aureus to actually generate disease. Here's a little joke of George Staphylococcus. It's a joke on George Stephanopoulos. 1 to 6 hours after consuming contaminated food, stomach cramps and diarrhea. It's gram-positive. It's not. It is non-modal. These are bacteria that are all over your skin, and so it's very easy to get them bacteria into the food. Now, what's going to happen is that the bacteria gets into the food, and so there's bacteria in our food all the time. There's no reason to eat sterile food, but you need to maintain the bacteria concentration at a fairly low level. Consequently, we want to keep hot food hot. That's going to keep the bacteria from dividing and pretty much denature all the bacteria that's present. In fact, we'll sterilize it. But then when you cool it down, bacteria in the atmosphere can land on this. You touch it, the bacteria are present, and if it's cool but not cold, room temperature, it can start to divide, and you can get enough of a mass. I start to think of it as the bingo point. You have to get enough of the bacteria present to actually start causing the disease. The prevalence of the disease typically mimics warm weather, and so it follows warm weather. Primarily, we're looking at things like picnics and stuff, where you make a batch of potato salad. It's got a lot of fats in it. It's got probably a reasonably neutral pH, not too acidic. Let it sit out for a while, and because there is bacteria present there, by allowing it to warm up to 80s or so on the picnic table and sit for a few hours, you allow the conditions to occur that will allow the bacteria to grow and replicate enough to start actually causing difficulties. So not so good. Again, keeping your hot food hot and your cold food cold. So in this case with Staph aureus, there are a whole range of foods that are easily contaminated. The staph is being transmitted because of food handling. And the bacteria can begin to replicate, and then the toxins that they can secrete can begin to generate high enough concentration they can cause problems. So this is typically going to happen when food is not being chilled properly. The toxin is heat stable, so cooking isn't going to destroy it, but the idea being that if you don't have sufficient load, you're not going to get sick. So by keeping hot food hot and inhibiting the bacterial growth, you can maintain it, or keep it at a low enough concentration. It's stuff that you typically would think of as being cold. Fresh cream cakes, milk and dairy products, salads and things like this, sandwiches, cold sandwiches. If they're not kept cold at a low enough temperature, or they're allowed to be at a warm temperature for a long enough time, bacteria can begin to grow, and you can get a high enough concentration that, treatment, stay by a potty. That would be pretty much the only thing you can do. It is going to be something you can clear fairly fast, and you're going to have issues, gastrointestinal issues, for 12 hours or so, and then it will subside on its own. Another Clostridium, but this is Clostridium perfringens, is another source, fairly common source of food poisoning. And again, this is an exotoxin that these bacteria produce, gram-positives, endospore-forming, anaerobic, and they can be present in the food, generate enough of the toxin that after a time of eating the contaminated food with a sufficient amount of it, you actually start to get sick. So cooked meat that isn't chilled real well before eating, that can allow spores to germinate. Things to think about when you're at a potluck. Great. Now, in terms of the GI tract generating different kinds of actual infections, we've got different kinds of bacteria now. Salmonella enterica, which is specifically the serotype known as typhi, produces something called typhoid fever. And typhoid fever is primarily found in areas that don't have very good water quality, they don't have good sewage treatment, poor sanitation. So, particularly with contaminated water. And it's one of the reasons why it's so critical for a public health standpoint to maintain a good water supply and to make sure that the sewage system is totally separate from the water system. And you would think, oh my God, this has got to always happen. There are a whole range of public health initiatives and processes that are maintained. Your plumber knows about these things, and there are whole sets of systems in place in plumbing to prevent what's called backflow. That is, you don't want to have contaminated water that's going down your sewer, and this could be like in your sink, from actually getting back into the water supply. And so all plumbing fixtures have things that are called backflow preventers. It's particularly important in places like toilets and things where it's a little bit harder to get into the water supply. But you could imagine there should be some strange ways if you don't got a real good toilet system. Like you'd have a hose that you're flushing stuff down, and the hose is in the contaminated material. Contaminated water could get back up and get back into the system in your house, and then you can begin drinking it. No, we don't do that. So by maintaining appropriate sort of plumbing technologies, you can inhibit it, but particularly by keeping the sewage treatment system separate from your water treatment system. This is a little point. One of the things that is done to test water, rather than testing for a range of different kinds of bacteria like salmonella or these other kinds of toxic bacteria that might be dumped out, as we see the bacteria that cause cholera. Rather than testing for those, a standard thing that is done to test water quality to make sure that it's safe is something that is known as a fecal coliform test. And that E. coli is the primary bacteria that is going to be in feces. And we can use that sort of as an indicator, because by itself it's actually not very toxic. But if you have fecal coliform in the water, that means that somewhere in your water system you've got a cross-contamination, or you're not processing the water properly, because theoretically you could take, and in fact, in actuality, you take the effluent from the sewage treatment plant and you can treat that, and you can actually have that as potable water. Now, not to be overly dramatic about this, but in the city of Decatur, we have the sewage treatment plant putting its effluent into the Sangamon River. The Sangamon River is what's going into Lake Decatur. Now, in the case of the city of Decatur, they're not dumb. The sewage treatment plant is downstream of the lake, but Urbana is upstream of the lake. So you have the possibility of effluent getting into the water that is going to be used as the drinking water supply. So you have to purify that. If for some reason the water purification system has failed, something's not going right, they don't have enough chlorine or whatever they're doing to sterilize things, you can end up with fecal coliform. And so rather than looking for a very specific bacteria, you use a generic kind of bacteria indicator. And so the water treatment plant here is every day carrying out fecal coliform tests. All right, so with Salmonella, it is a classic, as we say, oral fecal story. The bacteria is consumed, the bacteria multiplies, it irritates the body and the lining of the colon, and the bacteria leaves through diarrhea. Now, diarrhea actually is an interesting physiological phenomenon that we've been talking about some of the mechanics behind it, and we'll get into this in just a moment. So, the bacteria are acid resistant, meaning that they are able to survive the passage through the stomach into the small intestine and large intestine. Salmonella produces an enterotoxin that over time can begin eroding the intestine, and meaning that if left untreated, you generate bloody diarrhea as the lining of the intestine is being attacked. The bacteria can then get into the bloodstream, and that's going to be a very serious problem. And so you have the potential for a perforated bowel, small intestine, and that can cause dramatic infection, because your bowel really needs to be maintained intact, because there's all kinds of stuff in there that you don't want to get into the abdominal cavity. If it gets bad enough, you can start to run a really high fever because of the endotoxin. Pretty high mortality rate, 15%, due primarily to this infection in the peritoneum. Okay. So, just as an example of the spreading, there can be people that have chronic salmonella infections, and they can transmit this. You may have heard of someone known as Typhoid Mary. This is, in fact, a story about a cook who was passing the salmonella bacteria on, and in the food handling, and mainly she should not have been a food handler. She was arrested and quarantined for 3 years, as the story goes, and released upon the promise that she would not work as a cook. What did she do? She returned as a cook in 1915 for a hospital. Oops! Resulted in 25 infections and a death, quarantined for life. And post-mortem, it was identified that she had bacterial colonization in her gallbladder, which was maintaining a constant supply of the bacteria, and for whatever reason, she was somewhat insensitive to it, and she may have even been infected at birth. So people that have the bacterium can be passing it on. If you're maintaining proper sanitation, then it really shouldn't be too much of an issue. Another bacterium, Shigella. Shigella is a bacteria that produces what we know as dysentery. There's Shigella dysenteriae. In general, a disease called shigellosis. And it's gram-negative. It is present primarily in areas that have poor water sanitation, and we have the standard oral fecal route. About the only thing you do here is treat with antibiotics and hydration. And these are bacteria that can invade the intestinal cells and then actually begin reproducing and actually begin damaging the epithelial cells. Another bacterium that is causing a variety of different kinds of diarrhea and general sort of food poisoning effects is a bacterium known as Vibrio cholerae, and this produces the disease that you perhaps have heard, that is cholera. Cholera is again present in areas with poor sanitation. So cholera outbreaks happen when we don't have clean drinking water and clean water for washing. Typically sudden onset. You have phenomenal diarrhea, something that's known as rice water stools, which I hope to never have to even talk about ever again. This refers to the shedding of the intestinal lining. And death occurs through dehydration. Dehydration is going to lead to electrolyte imbalance, and your potassium levels can get completely whacked off, and you end up having cardiac arrest, all kinds of things. You can treat this by proper hydration. You know, people think about Pedialyte. As little babies get diarrhea, hopefully not from cholera, the electrolytes can get thrown off. And so maintaining appropriate salt balance is really critical. Let's see if we have another picture here. Well, just as an idea, in Haiti, after the earthquake in 2010, there were outbreaks of cholera. And it is something that happens and spreads very quickly. When people have that kind of diarrhea, it tends to splash. It gets everywhere. It's a mess. Oh, God. And it is a pretty serious problem when you have a lack of sanitation and lack of clean water. So like different refugee camps and areas that suffer a loss of their water treatment system, like in a earthquake, water pipes are breaking and so everybody's water is shut off. That's when this can start to happen. So the cholera toxin is an interesting game in that the toxin binds to the cell surface and it is going to lead to the release of chloride and sodium and potassium and then water into the intestinal lumen. All right, the inside, basically where your poop is. So just one of the things to think about here. Let's see what's next. Okay, all right. So you know from A&P that the bulk of digestion of the food is occurring first in the stomach, which is breaking stuff down through the action of acids and squishing stuff around. And then the food is slowly released into the small intestine. Now, the small intestine is doing all the heavy lifting in terms of nutrient extraction. This is where all the good stuff in your food is being pulled out. And what you're left with is material that you really can't digest. That's going to be fiber, cell wall material, bone, whatever it is you're eating, dirt. And this is material that you can't digest. It's going to be fairly liquidy. In fact, really quite liquidy. A lot of water in it. It gets into the large intestine, and the large intestine has one job, and that is to slowly begin pulling the water out of what I think of as the poop to be, because if you dump it out in the small intestine, whoosh, it's all liquid. The large intestine is pulling water out. How does it do that? Glad you asked. It does this through osmosis. Remember, we were talking a little bit about osmosis and the swelling of cells due to salt concentrations bringing the water in, and then because bacteria typically have a cell wall, they're able to maintain pressure. But our cells don't have that. Regardless, the key point here is that water always follows salt. You can't pump water in and out of a cell, but you can pump salts in and out of a cell. So if you want to move water around, you move salt into that area. The higher the salt concentration are these ions, sodium, chloride, potassium. The higher the salt concentration, the more water is going to be drawn to that area. So you don't move water around, you move salt around. Water always follows salt. I always say that's one of our camp cheers or mantras. Water follows salt. Wherever the salt goes, the water will follow through osmosis. Now, the way the large intestine works is it's got a variety of ion pumps that are pulling the ions out of the poop to be. Pull the ions out, the water follows, and you end up with a nice, dry, firm turd. If you've got a dog, you know how important that is. If you're a human, whatever. All right, so if you don't pull the salt out of the poop to be, you can't pull the water out. And because the cholera toxin is dumping salt into the poop to be, it's now going to allow for water to be pulled in as well. So you got a double whammy. You're losing lots of water, plus you're losing a whole range of electrolytes that you need to maintain for electrolyte balance. So this can be very serious very quickly. One of the key issues with diarrhea is, in fact, electrolyte loss and dehydration. So the first thing you start is if a patient has diarrhea, you got to get them on an IV and start pumping them full of liquids. And you also have to get electrolytes up, maintain their sodium and potassium levels up. Just as a little point then, we can use salts as laxatives, particularly salts that are not very well absorbed by the body, particularly things like magnesium. So magnesium-based laxatives, things like milk of magnesia or the colon prep solution they use for colonoscopy. It's got a lot of magnesium in it. The body can't pull magnesium out particularly effectively. It's just not one of the ions that we tend to see in our foods. And so high concentrations of magnesium stay with the poop. And as it makes its way through the large intestine, that high magnesium concentration is sort of offsetting the chloride and sodium that you're pumping out. And what happens then is that even though you're pumping out the salts that you normally would pump out appropriately, you still have a high concentration of magnesium. That's drawing water back in, and whoosh, away you go. A magnesium-based laxative. Okay, so that's a problem. A very common bacterial contamination is, in fact, our friend E. coli. Now, most E. coli are not toxic. Even at reasonably high concentrations, they're not going to be a problem. But some E. coli, specific strains, actually will have DNA from either a virus or other kinds of sources. And we have what's known as the toxin-producing plasmid. And this is what gives rise to specific strains of E. coli that are, in fact, very toxic. And meats can be contaminated with specific strains of E. coli that can be toxic. Now, probably most meat has a low level of E. coli in it. All right, not to be overly graphic, but you have a cow going into a slaughterhouse, and the cow is killed. It is immediately hung up, the carcass is, and they begin opening it up, pulling the guts out. And I've never been to a packing plant, and I really don't want to go, but these guys are good. They're pulling the intestinal material out without rupturing it. I mean, if you've ever done any deer hunting, you know that a gut shot can contaminate the meat. And so you want to go for a head shot or a lung shot. And then when you're dressing the deer, you very carefully remove the intestines and the stomach. You don't want to spoil the meat by rupturing those things. So in the case of large-scale cattle processing, there is going to be the occasional mix or oops or splitting or God knows what, I don't even want to know, of fecal material getting into the meat. And if we've got the appropriate E. coli strain, they've got a couple of things in them that make it so that they are very toxic. Lipopolysaccharide synthesis can be on these plasmids. Hemolysin, which is going to begin breaking down red blood cells. And then even nastier, antibiotic resistance. So you can have a whole range of antibiotic resistance on these cells as well. They can produce adhesion proteins that make them very sticky, and that's why in the meat processing business they are constantly checking for bacterial contamination and specifically looking for toxic strains of E. coli. When this happens, the meat is typically recalled. One of the things that happens, I've read that during big major meat recalls, actually only a very small fraction of the meat is actually recovered. The rest of it's out in the population. People have already eaten it, and generally they're not too sick, but occasionally people do eat contaminated meat and succumb to it. They can actually die from it. So these are specific issues that are a concern. All right, we're going to stop here with Helicobacter pylori, which is actually a really interesting bacterium that we now know is responsible for ulcers. And we'll pick up on this on Wednesday. So make sure you're cooking your food, keep your hot food hot, your cold food cold, and drink clean water. And good luck. Hopefully talking about diarrhea doesn't give us diarrhea. That's all I know. Hey, you guys take it easy. I will talk with you next on Wednesday. Bye.